Welcome, everyone. We're delighted to have you join us today for a webinar from Wave Life Sciences, who'll provide an update on the FORWARD-53 study of Wave N531 in patients with Duchenne muscular dystrophy. This is an exciting and pivotal time for the company as they share this 48-week update, as it is expected to lead the way to filing a new drug application for accelerated approval for Wave 531 next year. I'm Michael Kelly. I'm the Chief Scientific Officer at Duchenne, and I'm going to get us started with a bit of housekeeping. We'll have an opportunity for Q&A today. You can click on the Q&A button at the bottom of the screen and submit a question anytime throughout the presentation. There will be a moderated Q&A at the end of the webinar, but we may not have time to answer all questions live. For those left unanswered, we'll work with the Wave team to follow up with you after the webinar. After today, this webinar will be available on Duchenne's website as a resource for the community, in case you would like to share it with others. So let me begin. Wave has pioneered the design of novel stereopure oligonucleotides based around a phosphorylguanidine-containing backbone. This technology provides improvements in potency and stability and allows the company to target the genetic cause of a disease. To talk more about the platform, an update is on the remarkable 48-week results from the FORWARD-53 study. We've Dr. Paul Bolno, the President and CEO of Wave Life Sciences. So without further ado, let me hand it over to Paul to get us started.
Thank you, Michael, and thank you to Deb and the entire team at Duchenne for today's opportunity to speak with you. Some brief housekeeping opportunities from our side is we will be making forward-looking statements on this call, and as you are probably aware, this is an investigational medical product, and so we will continue to provide updates on the science. But to not forget why we're here, we are here and do everything that we have done over the past now over a decade at Wave because of you, the Duchenne community. You have been actively involved in our science. You are why we get up in the morning to do the work we're doing, and we're excited to share with you the work that we continue to do as we advance the current portfolio. So why, as Michael said, what we're here to do today is to talk about WVE-N531. And importantly, while we'll talk about the 48-week data, I think it's important to remember why we're here and how we got here. And how we entered this space with N531 was really through a robust amount of preclinical work. So we got to these clinical data by trying to step back and really understand what it was unique about our designs, our oligonucleotide chemistry that was getting us better muscle accessibility, better potency, better durability, and better retention. And that means that durability, meaning the potential for infrequent administration and dosing, but also the ability to get into different parts of the body. So getting beyond skeletal muscle and being able to think about exposure to heart and diaphragm. We did a lot of our work while early on in the MDX model. We felt like the double knockout model was an important preclinical precursor to moving into clinical trials. And we felt that this was important because this particular mouse that you study on only has dystrophin knocked out and doesn't have any compensatory proteins put in, meaning utrophin. Meaning the effects that you see in this mouse model are purely for the restoration of dystrophin. What was remarkable was not only do we see high levels of dystrophin restoration in the skeletal muscle, but we actually see higher levels of dystrophin restoration in important areas like the heart and the diaphragm. And this conferred in this model where there's usually a mortality at 8 weeks, so it was a rapidly progressive Duchenne model. We saw 100% survival at the end of the study at 40 weeks. So meaning that we were not just restoring dystrophin functionally, but it was actually improving benefits in terms of cardiorespiratory. And in fact, we actually saw in these mice the ability to restore respiratory function back to wild type levels. So this was highly encouraging as we thought about translating these findings into a clinical study. We also did work in the non-human primate, the NHP, and what this let us do is be able to again confirm that this exposure, so that the drug was getting not just into muscle, but actually at higher levels was getting exposure into heart and diaphragm. And so these data continued to be highly encouraging preclinically as we sought to move into the clinic. Now, FORWARD-53, as we are advancing, it had several important components to it. And that first important component was an exploratory experimental study, meaning we had just demonstrated these data in mice and in non-human primates. And in moving into early clinical studies, we wanted to see could we translate these similar findings into our patients. So part A was a small study. It was done in 3 boys where we had 3 doses over every other week, so 6 weeks in total. So a very early study to look at the improvements that we could see with this medicine. What we saw, which was pretty remarkable, is we were achieving high levels of skipping. We had 53% skip transcript, and we had shared these data on a previous webinar. We saw very high levels of muscle tissue concentrations at 42,000 nanograms per gram. To put this into context, we're talking about muscle tissue concentrations about 20 to 30 times that's what's seen with conjugated delivery. So without a conjugate, the chemistry was delivering high levels of muscle tissue concentration of medicine, generating high levels of skipping. What's unique, and we'll talk about how these findings will continue to persist over now a year, was the distribution not just to the muscle fiber, and there's a lot of discussion about delivery to the myofibers, but very uniquely, we were also delivering to the muscle stem cell. So if we think about, and we'll share more about this, what happens as muscle becomes injured and tries to repair, is that the stem cells that are within the muscle tissue become activated. And by restoring dystrophin there, you could potentially see continued improvement in muscle. And so it was very encouraging at this very early time point of 6 weeks to already see that we were getting exposure into this unique compartment within muscle, meaning the muscle stem cell. Additionally, and importantly, it was safe and well tolerated with no SAEs. As we continued to think about the progression of the study, we shared these data last year, which was the 6-month, the 24-week interim analysis of FORWARD-53. And this is continuing that same dosing regimen, so 10 milligrams per kilogram every 2 weeks, but now in an expanded patient population. So we had 11 patients, 10 ambulatory boys, and we had 1 non-ambulatory boy that was in the study. What was unique at that time was we were now able to see the translation of what essentially was saturation of muscle. So we had lots of drug in the muscle, and that was sustained. We saw skipping was sustained. So again, we reached a plateau phase on the exon skipping, but now it's translating to protein production. And we saw that very importantly, and we'll speak to this more in the 48-week study, is the consistency with which boys had elevated levels of dystrophin above the Becker 5%. And we had a mean of 9% dystrophin. It was safe and well tolerated, again, with no SAEs, important as we think about moving this medicine forward. But now what was unique is in addition to getting into those muscle stem cells and muscle myofibers, was we saw a shift in the muscle tissue as we could look at it. So these are on the muscle biopsies to what essentially became muscle fiber regeneration, meaning as we were talking about that activation of the stem cells. What we now saw was that they were getting into the right compartment of the cells and actually turning on those regenerative cells to actually start regenerating muscle fibers. Now, importantly in the study, and what we would expect to see as leading indicators of improving muscle, was we saw an improvement in muscle biomarkers, including CK levels, meaning that reparative process was underway. What we also saw was a 61-day half-life, meaning the profile of this medicine looks to be monthly, amenable to monthly dosing, meaning high levels of muscle concentration. It's retained in that muscle fiber for a very long time, and that would allow us to dose much more infrequently. So it was really next for us in thinking about this 48-week data set where we've shown high concentrations, we've saw high levels of exon skipping, high levels of protein production, was really to see, does this now translate beyond protein production to actually signs of improvement in muscle health and functional outcome benefits? And indeed, that's exactly what we saw. So the update we're going to share now is on the 48-week data set. So what we did see at our 48-week time point is that WVE-N531 remains safe and well tolerated with no serious adverse events. We continue to see consistent dystrophin expression averaging about 7.8% between the 24 and 48-week time point, with 88% of the boys on the study above 5%. This is important because it means that we're now reaching a stabilization of dystrophin production in addition to concentrations and exon skipping. Now, what's unique in what we continue to see is that we saw statistically significant reductions in muscle fibrosis, CK levels, and these were driven by decreases in inflammation and cell death. And we'll share with you some of the histology, the evaluation of those muscle fibers, where we can actually see this take effect. And what we saw also was this transition from a regenerative state in the muscle, so kind of fixing itself in repair, to more mature muscle. And so this is the continuum that uniquely suited from the study we're able to do, where we have biopsies at 6 weeks, 6 months, and now a year, is really to follow the progress of what regeneration to maturation looks like over time. Now, moving beyond dystrophin levels and biomarkers is that we did see a statistically significant and clinically meaningful improvement, 3.8 seconds, in time to rise versus natural history. We'll talk more about that, and functional benefits on other measures, including North Star. If we step back and look at the boys on this current study, is we did have boys across age range, and that's important. And we heard a lot from the community about thinking about ages for patients on study between 5 and 11. And the remaining part of the characteristics are pretty standard. We did see all boys on stable doses of steroids, and that's going to be important as we speak to the improvements we're actually seeing in muscle inflammation and fibrosis, that these were on sustained stable levels of steroid. From a safety perspective, all events were mild to moderate. We had one severe that was non-drug related, just to put that in context, because I know people look at that. That was a case of viral gastroenteritis. It was thought to be secondary to norovirus, and it did resolve. So starting with the dystrophin levels within the study is we did see this stabilization between the 24 and 48 week time point. These differences between the 24 and 48 weeks, as you can see, stable across skipping, but importantly on dystrophin, these are within the ranges of the Western blot assay. And so we did other orthogonal studies that we did share on the earlier update call that actually confirms that the 24 and 48 week data were similar. And what this did was allow us to average that dystrophin levels across to really think about the stable levels of dystrophin of about 7.8%. What's very important, that's on the right-hand side, is looking at that proportion and that distribution across patients. Because we know one of the important features of dystrophin restoration is seeing that that's consistently across patients on the study. And therefore, being able to think about the benefits that are conferred, those are conferred by having consistent levels of dystrophin across boys on the study. Now, when we talk about muscle health, regeneration, we need to step back and say, well, what happens in healthy muscle? And in healthy muscle, when there's injury, that stem cell pool becomes activated. So they're in the muscle for a reason. They're there to respond very quickly to a sign of inflammation and help restore and repair muscle fibers. So the stem cells become activated, they become new muscle fibers, and they continue to build and help restore muscle back to its normal function. Now, as many of you may be aware, when we talk about DMD and the muscle and fibers and the stem cells lacking dystrophin, those stem cells begin to turn on, but they're inefficient to repair and become new muscle fibers. And over time, that leads to fat and scar tissue, decreasing the repair mechanisms within those cells. And if you look at that cross section, those cells are over time are unable to repair and again become filled with those fat and scar tissue and then unable to function. What's unique in the study is because we were able to look over time, and these are the biopsies across all of the boys who had biopsies at the 6 month time point and the 1 year time point at 48 weeks, is we're able to do cross comparisons of each patient on the study and to be able to look at the improvements that could be seen in muscle fibers. And what you notice right away, and you'll see these little purple dots that located that are infiltrating throughout the muscle, is those are the inflammatory cells. So what you see is at that 6 month time point, the muscle fibers are starting to repair. That's what we call that regenerative cycle. But as you look out over the 48-week time point, we see a reduction in those inflammatory cells. We see an improvement in muscle fiber, and we see less scarring. And we'll walk you through these data over time. But at a very large level, when you step back and look at the health of the muscle fibers between that 6 month time point and the 1 year time point, we continue to see improvement, not just in one patient, but in all the patients on the study. We also tried to quantitate this. So the last slides were images. We could look at measuring cells and structures, but we also could look at quantification of moving from, on the left side, inflammation between 6 months and a year, with a shift in patients to a more minimal and mild range of inflammation. And on the right side is cell death. So this is necrosis. These are cells that become injured and eventually die. And what we could see is an actual reduction in cell death as well between the 24 and 48-week time point. Now, the improvement in muscle also translated to what we would expect to see, which is a reduction in CK levels on the left side with baseline, and continued improvement over time. And on the right side versus a DMD natural history cohort from CPATH, where we could look over time at improvements in CK changes on boys on the study, as well as the natural history cohort. So we continue to see CK levels decrease. Now, beyond looking at measurements like CK, it's important for us to see, well, what's driving that improvement in muscle fibers? And what we could see on the left-hand side is what you'd expect to see as muscles get healthier is a reduction in the density of stem cells, meaning the repair, you need less cells to repair a given section of tissue because muscle's healthier. And so therefore, those cells can actually go in their resting phase to be there to react. On the right-hand side, we could also be able to look at a very important measurement of a healthy muscle fiber, which is the reduction of internal nuclei. So as muscle cells mature, those nuclei move from being inside the cell or internalized to being more peripherally located. And what we saw was exactly that change between the 24-week time point and the 48-week time point, meaning that the muscle cells themselves were becoming mature. Now, what was remarkable and hasn't been seen before is that does this translate into a reduction in fibrosis? I mean, actually, are we reversing some of the muscle damage and injury over time by treatment? And we could actually see across all of the boys in the study that could be evaluated at this time point that we did see that, a reduction in fibrosis between the 6-month time point and the 48-week time point. On the left-hand side, being able to plot that. On the right-hand side, the image generation that helps generate these data. So these are quantitative data that are statistically significant in terms of that reduction in fibrosis and really demonstrates that we're seeing the shift in dystrophic muscle towards healthy muscle, which as we know, as we think about exon skipping and importantly, what dystrophin restoration therapies are supposed to bring about. It's supposed to be able to drive that repair between dystrophic muscle towards healthy. Now, as we think about all of these data in succession, meaning we get drug to where it needs to be, it's doing what it needs to do in creating dystrophin by creating dystrophin in both the myofiber and uniquely for Wave in the stem cell. We see that generate regenerative muscle, but then we also see this mature muscle. The next step that we wanted to see was, would this ultimately translate to an improvement in clinical function? So to be able to do this in the study, we had a very well-matched natural history cohort. This is a 53 cohort, so we had a very good matching between the patients on our study and the patients in this cohort. And what we could see was a significant, both clinically and statistically significant improvement of 3.8 seconds in time to rise. So this is very important as we think about not just looking at what's happening in a single muscle fiber through the biopsy, but really what's transitioning functionally in terms of measuring and seeing does this improvement in dystrophin, the improvement in muscle repair and regeneration ultimately now translate into a benefit in function. Additionally, when we look at the North Star, which we know traditionally takes more time, and we look at the same match cohort, we do see an improvement, and we do expect over time, and we'll talk about how the study is continuing, to continue to follow the boys in the study over longer time periods to look for continued sustained improvement along the North Star. We also looked at more peripheral measurements in grip strength, and we'll continue to follow these over time and did see improvements from baseline on grip strength. We continue to receive feedback from patients on the study and the clinicians who've been involved in terms of their own reports of what they've seen. And it's highly encouraging, as any study is in this disease, to see improvements noted from clinicians. I like to see this feedback in a setting where we also see on histology changes, dystrophin changes, and clinical outcome changes, that there's a good correlation between what we're hearing and what we're seeing as part of the clinical trial. As we think about the profile of this medicine going forward and where we see advantages, we do see the importance of consistency. So we see not just levels above 5%, so again, 88% of the boys above 5%, but we see that consistently across the patients, which is going to be important as we think about continuing to drive treatment and improvements. We do see uniquely the evidence to get to those muscle stem cells, those regenerative cells in the muscle. And based on those exposures, concentration, and what we see from durability, we do believe that this will be a monthly medicine, monthly dose medicine. We have a favorable safety profile. We see favorable changes on clinical benefits. So now starting to translate again dystrophin to clinical outcome benefits, with the first evidence of disease reversal in muscle with dystrophin restoration. And as we shared uniquely preclinically, the unique ability and evidence to reach heart and diaphragm. And that lets us think about the opportunities in ahead of us where we can think about, particularly in the non-ambulatory setting, and chronic treatment to make sure that we're improving cardiac function as well as respiratory function over time. So what's next is we continue to think about moving both N531 forward for exon 53 amenable boys, but being able to think more broadly. We do believe that the current portfolio as it stands would address up to 40% of the DMD community. We do think continually about other exons that are amenable, and we'll continue to look for opportunities of how best to present that with a focus near term on advancing Forward 53. So all boys in Forward 53 have consented and are now receiving monthly doses of N531 in the extension portion of the study. We are going to expand Forward 53 to include additional boys on a monthly dosing regimen. We do, as Michael said on the call, intend to submit a new drug application in 2026 to support accelerated approval with monthly dosing. And we are encouraged to be engaged with the agency as well as more broadly as we think about outside the United States, on the opportunities for what the global confirmatory study will look like. And it will be a global confirmatory study. Beyond N531, we are advancing and have programs for exon 52, 51, 45, and 44, and we expect to submit multiple clinical trial applications for these other exon skipping candidates in 2026. As we think about the data that got us here, we do reflect on all of the engagement that we've had with the community over time, and would really like to spend a special thank you to the boys, families, clinicians, study site staff, who we know do a lot of work to get us to where we are, who participated in the study. And we thank all of you for spending time with us, whether it's this morning or this evening, on the webinar. And we're happy to spend time asking questions or answering questions.
Paul, super well done. That's a really, really nice, inviting data set that's been generated in patients. And I like the way the whole thing has come together. Let me invite our VP of Research, Dr. Liana Orlando, to join as well for the Q&A session. Hey, Liana.
Hello,
Hey,
everyone. Thanks,
Yeah.
Mike.
Yeah, but one thing I would kick off with, Paul, is like from my perspective, it's the totality of looking at reductions in inflammation over time, the change in muscle from dystrophic to a more healthy state, markers for regeneration, and the reduction in CK. All of that, along with the amount of protein that you're putting in there, is really compelling. Congratulations to you and the team, and that's a really nice data set.
Well, thank you, and we appreciate it. And there's more data, and we're cognizant of data with the community, but in addition, as you pointed out, I mean, reductions for those who are familiar with inflammatory markers like IL-6 and MCP1, I think there is a substantial amount of totality of the data on this reduction of inflammation that is highly encouraging as we think about being able to see the improvements over time. So yeah, it's nice to see a comprehensive data set and muscle improvement.
One thing for me is the data was all generated on Q2 week dosing, and you're now moving to Q4. Are you expecting everything to stay the same? Is there any further looks at data at that time? Because that's going to be the information that the agency will look at for accelerated.
Well, we've had the conversation with the agency with actually the 6-month data. So that was the biweekly data. So we do have a path, as we talked about, through dystrophin on the 6-month data set. But what's unique, and I think it's been highly consistent, is we are getting exposures that are basically saturated muscle, and that stays over time. So what we're not seeing is this kind of waning of drug concentrations, waning of skipping, or protein. And I think that was another piece of what was encouraging, is that the concentrations are staying, the exon skip transcript is plateauing, and then dystrophin is stabilizing. And I say stabilizing, not plateauing, because I think we don't have continued biopsies to see what's happening next. Is it that muscle fibers are becoming stable and therefore drugs going into other fibers and what's happening on a dynamic basis? But all of it suggests, again, with the half-life in the tissue of 61 days,
Mhm.
going to a 30-day dosing regimen doesn't have us losing exposure. But actually, I think beyond just convenience, I think it's the right thing to do in terms of optimizing both dose and frequency going forward. And I think for families, as we've spoken to many of you, the shift to monthly without losing efficacy is important. And I
I
think
totally agree. I think it's a great additive to the target product profile that you're generating out there. What's your thinking and plans as you look ex-US, for taking 53 forward?
yes, I mean, as we said, the engagement with regulators had historically been based on our 6 month, which was dystrophin data. I think, as we know, not just in the US, but in particular outside the United States, clinical endpoints become really important in those discussions. And so we're excited to take, obviously, the package of data now, where, as you pointed out, we've got the dystrophin discussion coupled with muscle regeneration and maturation, and then clinical data, and be able to have the conversation outside the United States too with regulators around clinical endpoints and the appropriateness of the clinical data that we have, and how that could support, further studies, meaning the confirmatory study, but also what registrational pathways could look like now predicated on clinical data sets.
Mhm. Leanna, you got some questions there?
Yeah, there's you mentioned that you're looking at a number of different other exons to skip, and that the sorry, the timing on that would be, I think you said looking at filing CTAs next year. Was that correct?
Yes. And I think, one of the areas that we're in active exploration, and particularly in with US regulators, is umbrella studies. So, where we can think about platform design and studies. We had done this in the past. We had an exon 51 program that we had run an extensive late stage study with engagement around regulators on umbrella design. So this is not the first time that we're engaging, but we're excited now with the data we have to have a productive conversation on what an umbrella study is. And for those not familiar with what we mean by saying an umbrella study, is it's a way for us to look at a number of exons where we can think about using common shared placebo arms. So recognizing that within a variety of deletions, there are very similar background levels of dystrophin and very similar clinical characteristics. And in those opportunities, we can think about having one placebo arm that could support a number of exons in the development, really enabling, and I would say us, I think us in the broad sense of the community, to run highly efficient studies where we can minimize number of boys on placebo-controlled studies, be able to optimize for treatment, and be able to run those much more efficiently and effectively to a dystrophin outcome and potentially a clinical outcome. And so those are the conversations now on the other side of this as we think about the design of the confirmatory study for exon 53. We do think about that design as being supportive from a placebo arm side of being able to support a number of these studies. And so that's the work that's underway right now in terms of optimizing how do we expedite getting into the clinic, but most importantly, how do we optimize those studies subsequently to efficiently deliver a data set?
Thank you. And you completely anticipated where my question was going. And, as you very well said, that's wonderful because not only can you minimize the number of individuals, right, who would be on placebo, but also you can be moving these programs forward, at the same time and more efficiently. And so, it's always exciting. You got to get the first one across the finish line. But since everybody's always asking about, okay, what's next? When will you get to the exon that I need skipped? It's really wonderful to hear just how much thought and planning is going in to actually to meet that desire. So I appreciate
No,
that.
absolutely. And look, this happens with partnerships. So, as we talk to you further, the community further, and other groups. I think, this is not just a wave develop design, right? How do we think holistically for the whole DMD community? How do we all expedite these clinical trials faster, more efficiently, and with high quality? So I think, it's not sacrificing the quality of the clinical trial to efficiently design placebo arms so that we can expedite development. So it's something we're excited about working on.
Yep.
Um, I have one other, it's very future thinking. So, but since you said you're really getting saturating levels of your drug in muscle, I was wondering if that enables you to sort of think about being able to put in combinations of oligos to be able to start thinking about multi-exon skipping, or this is another thing that, individuals in our community that really that's what they need. They're always asking about.
It's a wonderful question, and it's something we've spent a lot of time thinking about. One, the exposure level was, what we've seen and what we anchor on, which was delivery from first and
Mhm.
foremost. I think that's important as we think about muscular dystrophies and diseases within muscle more broadly. So getting accessibility is key and paramount. We have actually spent time thinking about multi-skipping, and it's something that we've thought about various designs on. I'd put it in the scope of early research
Mhm.
because there are complexities with it in terms of delivering multiple exons that are already formatted and made, absolutely. But we have thought about the complexity within splicing and are there opportunities to think from a research perspective and then ultimately a development perspective. And as you well pointed out, the first step always for this is deliver the complete data set. I think right now the goal has been, can we show that we can restore dystrophin and do that safely? Can we do that in a way that regenerates muscle and creates healthy muscle? And then ultimately, does that translate to the clinic? And by connecting the dots on being able to do that, then lets us, as we said, be able to develop other exons for, or exon skippers for other deletions, but also then continues to support that overarching premise from the discovery side of how do we think about the more complex opportunities that exist to drive kind of, as you pointed out, multi-skipping and being
Mhm.
able to think about that opportunity. So it does all build on itself. It all takes time, and we know that time is important to patients. And so we are working with a high degree of urgency, but we do realize that the first step is to validate from beginning to middle to end of demonstrating the clinical translation of dystrophin. And that starts with N531 and continues to build on from there.
Mhm. Thank you for that. That came from one of our good friends, Tushar, whose sons have got mutations in a fairly awkward region. And I think, not to speak to Tushar over the webinar here, but as that mouse model becomes available, that there's something that we may be able to actually utilize at some point with this type of technology. Paul, let me just ask a question on combination therapies. I noted in the patients that you used a combination with steroids, basically prednisone and deflazacort. Are you considering adding patients in that are on vamorolone, for example? Or what's your thinking and attitude around Duvyzat, which is now, getting more of an uptake in the community?
Yeah, I mean, this study was predicated on patients came on there
Mhm.
on a stable level of steroids, and we continue the study. I think it was interesting is the balance of the patients on both. So it actually, I think from being able to look at the clinical trial perspective and seeing a nice balance and that patients didn't have to change their steroid levels, I think that was highly encouraging. I think as we think forward, there's always the opportunity of, what I would say the inclusion are, because it's not a combination study, it's what are patients stable on and how can we just assure that in running the study, we can test the benefit, which is always the important thing of what a clinical trial is designed to do is what is the additive feature of what your medicine is doing in that clinical study. And so in this case, it was based on stable level of steroids, and we'll continue to see what medicines patients are on and stable on to be able to then to see what happens when, they're included in the study and we can see the impact of dystrophin restoration in that background setting. So at this point, we're still evaluating the utility of a variety of different medicines and what patients are on and how we can evaluate this medicine in the context of that.
Excellent, thank you. Liana, any other questions from your side?
No, I think we've pretty much covered it.
Yeah, I think I'm there too. I think I'm going to wrap things up, but I feel that this really has, moved significantly in a short period of time. I know, Paul, you and I have talked about this over many years when you had the original 51 targeted program, and for the company really to step back and rejig and actually get to this point is quite remarkable. Many actually don't do that there. So I offer an enormous amount of thanks and congratulations to you and the whole team out there. I want to also thank everybody for joining us today in this webinar. And I think obviously the Way of Life Sciences team for everything that they've done developing this molecule. and for sharing what is a really important update on all of what we're witnessing in here with this type of chemistry, performing quite well. We look forward to further updates as we move closer to the new drug application for the US accelerated approval pathway next year. Post-webinar email will be sent tomorrow should you have any additional questions. And we'll get in touch with the attendees that were on today and let them know when the webinar is posted online for review and sharing with others. And once again, thanks for taking time to join us. It was an early start today, but have a wonderful rest of your day, no matter where you are. Thank you all.
Thank you.